Starting with this comparison of mitosis and meiosis, we're going to look at how these two processes are somewhat different. Notice in mitosis that if I started with a cell that has 4 chromosomes, notice in this case they are 2N. That is I have 2 sets, one red and one blue for each. And in mitosis, they would just line up and they would separate into separate cells with the daughter cells looking pretty much exactly like the original cell, except for possibly the cytoplasm. So in terms of DNA, they would be exactly the same. Now in meiosis, of course, because of the pairing of the homologous chromosomes, the first division looks quite a bit different because you're going to have let's see if I can get a little bit closer. You're going to have those dyads, and then the dyads are going to separate, and you're going to have cells that have half the number of original chromosomes. So that's kind of what we've already been talking about. So that's just kind of an overview. Let's look at some other things that are in comparison, all right? So first of all, we have some similarities. The similarities are that the replication process only happens once. That would be in synthesis of mitosis 1, or that is interphase 1, or in synthesis of just interphase if we're just talking about mitosis. I might have said that backwards. Meiosis would be interphase 1 and interphases of mitosis. All right, and then the other things are the physical thing that happens in each phase. That is like the spindle forming, the centrioles replicating, etc. All those things are going to be the same for each of the phases. So the phases generally and what happens in them is going to be the same. All right, so starting with some differences. First of all, in mitosis, as we already stated, there's only one division. So you end up with 2 cells. And in meiosis, you end up with 4 cells because there is 2 divisions. So that's one. The second thing is that in mitosis, any cell can do mitosis. That is, I could have a 1N cell or a haploid cell that if it undergoes mitosis, let's say I just had 1 chromosome. I'm just going to replicate it and put 1 chromosome in each of the cells. So I'm going from 1N to 1N. So the cells created in terms of DNA would be exactly like the cells that I started with. Or I could have a 2N, that is 2 chromosomes, and separate them. And they would have 2 in each of the daughter cells in mitosis. But I can only do meiosis with a diploid cell because the whole point of meiosis is to create haploid cells. So it's a reduction division, and it's going to go from 2N to 1N. And so I have to start with a diploid cell. All right. Now another difference, and this is kind of important because this is going to be related to genetics when we talk about it. So in mitosis, as we just stated, the DNA would be exactly the same. But in meiosis, there's a reassortment of the homologs. And we're not really talking about crossing over here. We're just talking about the homologs themselves. So let's say I started with a cell that had 2 homologous chromosomes. In this case, 2N equals 4. And I've labeled them capital A and capital B and little a and little b. So the homologs would be capital A and little a and big B and little b. Okay, so what can happen then is that when I just wrote the letters and the cells that are created. But if I have, let's say, capital A going one direction, then little a is going to go the other direction, right? Because I'm separating the homologous sets of chromosomes. And I could have, for the other set, I could have big B going with capital A and little b going with little a, or I could have little b going with capital A and big B going with little a. All right, so I could have either of those possibilities. So the way I figure out the number of possibilities is I always have 2 homologs that I'm separating, where N is the number of homologs. In this case, there's 2 sets. That is capital A, little a is one set, and capital B, little b is another set. So 2 to the 2 is 4 possible outcomes. So notice that each of those 4 are going to be different in terms of the homologs. Now, how many would that be for our 23 sets? That would be 2 to the 23. What is that number? If you calculate it right now, you'll see that it's about 8 million possibilities. So we're not even talking crossing over here. we're talking about the variation that occurs because of the shuffling or reassortment of homologs. All right. And then the last difference, of course, is crossing over. And that is something that we've already talked about.